sorted out here. So our first speaker, it's my pleasure to introduce Dr. Jonathan Lynch, who's joining us from Penn State University, where he is a professor of plant nutrition. Dr. Lynch received his bachelor's in soil and plant nutrition from the University of California, Berkeley, his master's and PhD in plant physiology from the University of California, Davis. And he's going to be talking to us today about roots of the second green revolution. So I won't take any more time away from you. Let's welcome Dr. Lynch.
Thank you, Gerald. Good morning, ladies and gentlemen. I'd like to begin by thanking the graduate students of Iowa State University for inviting me. As Kendall mentioned, I was very impressed with your organization and professionalism, and I'm impressed with you as young scholars and as people. So I'm very honored to be with you today. As Gerald mentioned, I'll be talking about roots of the second green revolution today, deployment of root fiends for improved water and nutrient acquisition. Here's where we began in the 1960s with rice and wheat. If we fertilized, we lost yield because of lodging. Norm Borlaug and others developed dwarf varieties that could respond to fertilization without lodging. This was the second green revolution. However, these varieties gave us no yield gain at low fertility. The question is, can we develop genotypes that follow the red line, where we have high yield with high inputs or good conditions, and better yield at low fertility? This then represents what we've achieved in the historical green revolution, and this area is the potential for a 21st century green revolution. Do we need such a green revolution? Here's some recent statistics. 850 million people hungry. That population is actually expanding in Africa. The number is not getting smaller, it's getting bigger. That translates into 3 million children dying from malnutrition every year. That's half of all childhood deaths in the developing world. And of course, the population is continuing to expand. It's expected to reach 10+ billion in the next several decades. Virtually all of that expansion will be in these food insecure countries. And in these countries, the single largest human occupation on Earth, in fact, is low input agriculture, meaning agriculture without fertilizers, irrigation, or pesticides. And of course, because this is low input agriculture, water and nutrients are strong limitations to productivity. Here's some data of productivity from different parts of the earth for maize, rice, and common bean. You can see the yield potential on the top for maize is 20 metric tons per hectare. In Africa, you're lucky to get 1 metric ton per hectare. So we're losing 95% of our yield potential from some sort of unfavorable condition. Paddy rice grown in water is still losing 80% of its potential yield in sub-Saharan Africa. Common bean, which can fix nitrogen, losing 90% of its potential yield in Africa. So there are many proximate causes to world hunger, but a principal pervasive cause of food insecurity is simply that we're not getting good crop yields because of environmental stress. And this is one of the reasons. Phosphorus availability is suboptimal for almost all life on Earth. This is a map of phosphorus availability for plant growth. Any kind of red color represents suboptimal phosphorus. Most of the Earth is actually suboptimal in phosphorus availability if we don't fertilize. Nitrogen. This is a map showing you nitrogen balance. So the green colors represent a positive balance. You can see places like China actually have excess of nitrogen in their systems, but sub-Saharan Africa is negative. So we're in a deficit. We're losing nitrogen every year. Drought, obviously, is the principal constraint all over the earth, and this is projected to worsen in years ahead as a result of climate change. The soil resource base is degrading. This is a coarse scale map from the United Nations showing soil degradation. You can see even the U.S. has significant soil degradation problems, primarily erosion. But in Africa, these problems are severe. The estimate was, in 2006, 75% of the cropland in Africa was degraded. And of course, this is ongoing, so it's worse every year. Well, what can be done about this? Integrated soil fertility management would consist of using inputs. I'll come back to that in a moment. Managing the soil in a mindful way to enhance and sustain soil fertility. Now, in my many travels in developing countries, you very, very rarely see these practices being used. In fact, my farmers in Pennsylvania, my neighbors in Pennsylvania are farmers, and they don't use these practices. So for whatever reason, land tenure or ignorance or lack of time, you don't see soil conservation practices being practiced in the poorest countries of the earth. And then, of course, the third leg would be adapted germplasm, which I'll be talking about today. But before we go on to talk about that, I want to come back to this issue of fertilization for a moment. This is a global map of fertilizer use on a log scale, and you can see the amounts of fertilizer used in sub-Saharan Africa are negligible. And in fact, every couple of years, the World Bank produces a new set of data, and we make a new map, and it looks worse than the previous one. Fertilizer use rates in Africa have been declining. One country we work a lot in Mozambique, right here, 90% reduction in fertilizer use over the past 10 years because of rising fertilizer costs. So in addition to long-term sustainability issues related to high-grade phosphorus ore deposits and those kinds of issues, it's difficult to foresee in the short to medium term how Africa is going to fertilize its way out of this problem. Which brings us back to the need to develop adapted germplasm. Now, the first story I'm going to tell you is with this plant, common bean, Phaseolus vulgaris, an important source of nutrients for about 1 billion people in developing countries. And this is a typical bean production system in the Andes. You can see from this distance, you mainly see the maize, but the maize-bean intercropping system next to this person's home. And if you look closely, the bean plants are highly phosphorus deficient. I expect this plant, based on its symptoms, to be losing 80 or 90% of its potential yield because of phosphorus deficiency. And this is typical for crop production systems in developing countries. So I began looking at this problem. I got my PhD and went to work at the International Center for Tropical Agriculture in Ciudad, Cali, Colombia, where they had been trying to find bean lines with better performance in low phosphorus soils for 20 years. We're 3 degrees north of the equator here, so they had 4 cycles per year, several thousand lines every cycle. So over 20 years, they must have screened around the order of 50,000 or more lines. And just as I was arriving there, they had written a couple of papers saying it was impossible to beat the yield of this Brazilian landrace, carioca. And they closed down their program, fired their soil scientist, and said, nothing can be done to improve phosphorus efficiency in beans. Carioca is the winner. Well, I didn't really like this conclusion. I thought there were several problems with this kind of an approach. For example, brute force yield screening, I think, is usually doomed to fail. We might have inadequate diversity. These were elite lines that have been selected for yield potential and disease resistance, and then as an afterthought put into this stressful nursery to see if they had phosphorus efficiency. So they probably were not sampling land races and so on that might have better adaptation to stressful conditions. There are confounding adaptations. With bean, the photoperiod and temperature is very important in determining yield. And so we might have a line that might have good phosphorus acquisition characteristics, but is not adapted to this particular location and will not yield well. We might have confounding stresses. In fact, in this particular field, they had compaction from driving tractors over this for 20 years. They had aluminum toxicity, manganese toxicity, low calcium, a variety of problems. So they weren't being very specific in the stress they were imposing, and that is problematic if you have distinct adaptations to distinct stresses. There are phene interactions, which I'll be talking about later this morning. That some of these traits, or what I call phenes, may have value only in certain phenotypic contexts. And finally, there's fitness trade-offs. We might see that a useful trait has a downside or a trade-off that may not be apparent when we just look at yield. So I thought rather than doing this kind of brute force yield screening for phosphorus adaptation, that we ought to take a more targeted scientific approach like people do for disease breeding or insect resistance breeding, where you have a particular stress and you're interested in particular biology in response to that stress, and you might find these resistance traits in different parts of your germplasm, but you as a breeder have to put it together in an intelligent way. And to make a long story short, we were able to identify a number of traits related to phosphorus acquisition. It turns out they were all root traits. And the key thing to point out here is that the topsoil is where you find phosphorus because it's highly immobile, so it accumulates in the topsoil over time. So root traits that enhance topsoil exploration will enhance phosphorus acquisition. And I'm not going to go through all these things. I'm just going to highlight a couple of examples to show you the kind of things we were seeing. This is one of the things we saw early on, which is the growth angle of the main root class in this species called basal roots varies. So these are plants excavated from the field in Honduras, and you can see these basal roots are fairly steep, and these ones, this genotype, fairly shallow. So this one will have better topsoil exploration. In fact, when we made recombinant inbred lines from contrasting parents and looked at them in the field, we saw a good correlation between phosphorus uptake and the root shallowness. In fact, we have about a 6-fold variation in phosphorus uptake caused by variation for this traits among lines with a similar genetic background. This was a simple QTL study back 10 years ago. We didn't have a very saturated bean map. But so here's the 11 chromosomes of bean. Red is where the loci associated with performance in low phosphorus fields in the tropics. And yellow, blue, and green are loci associated with root shallowness that we measure in a plastic bag in the laboratory. And you can see we have regions of co-segregation, which is good evidence that, yes, indeed, root shallowness is related to phosphorus efficiency in the field. Here's another trait. Now we're looking under a microscope, and we're looking at root hairs, which extend out into the soil and help the plant acquire phosphorus. This is the result of scientific breeding, and this is a landrace we found from the highland region of Peru. It has very long and dense root hairs. And as you might expect, the plants, once again, recombinant inbred lines descending from those two parents segregate for root hair length, and as you might expect, the longer root hairs assisted phosphorus acquisition from the field. Here's the field environment where this data came from. So we had weeds and nematodes and a variety of other problems faced by small farmers, and yet we have a very good, tight correlation. We can increase phosphorus acquisition about 250% by selecting for long and dense root hairs. So here's another trait that's conferring phosphorus efficiency that's under relatively simple genetic control. Now, one of the issues we've been dealing with in deploying these in breeding programs is how these traits combine or stack up, how we can develop an integrated phenotype using these traits. And in this particular case, I've shown you that shallow rooting is good for phosphorus acquisition. I've shown you that long and dense root hairs are good for phosphorus acquisition. Well, how do these two traits combine? Now we have a set of recombinant inbred lines that descend from the same two parents that vary for both of these traits. So if we have a set of lines that have short root hairs and deep root angles, this is our baseline in the field in Mozambique. That's what we're considering our baseline in a low phosphorus soil. If then we compare those to rills that have the shallow roots, these shallow rooted rills have 60% increase in growth in these low phosphorus soils. The ones that have long root hairs have a 90% increase in growth in low phosphorus soils. So if we have plants that have both of these traits together, we expect 90 plus 60, 150% increase in yield. But actually we observed a 300% increase in yield. And this is actually logical, because long root hairs will be more useful on shallow roots when they're in the topsoil than when they're on deep roots. And so I think this is an important concept, certainly for these root traits, that there are significant positive and negative interactions among the traits that we have to understand to put together an integrated phenotype. There are also trade-offs for these traits. This is shown in this data from the field in Honduras, where we have drought and low soil fertility. Once again, here's a set of recombinant inbred lines that vary for root angle. If we irrigate and fertilize them, they grow the same. If we put them under low phosphorus, the shallow-rooted lines do better, as you might expect. But if we put them under drought, the deep-rooted lines do better, as we might expect. And so this is, of course, a problem for breeding, because we don't want to produce a drought-tolerant line that only works in high-fertility soils, or vice versa. We want both of these traits. Fortunately, in this particular case, we were able to find a trait that helps us deal with this trade-off. We discovered that basal roots in beans appear from distinct positions. So this genotype has one position, which we're calling a whorl. Each whorl produces 4 basal roots because of the anatomy of bean. This genotype produces 3 whorls. And the more whorls we have, the more basal roots we have. So we can go from 4 basal roots to 16 basal roots by going from 1 to 4 whorls. And the idea was, the hope was, that having a greater number of whorls and a greater number of basal roots would give us a broader vertical range of soil exploration, so we could have topsoil resource capture as well as water capture. So these are the phenotypes. This is computer simulation, but these are what the phenotypes look like. 1, 2, 3, 4 whorls. Here's one whorl. Here's the second whorl, third whorl, four whorls. And you can see we have a broader range of exploration here. And this was confirmed in the field. This is work in Mozambique. You can see this red tropical soil. We had a 60% greater growth at low P comparing 2 and 3 whorl genotypes from the same genetic background. So a significant increase in growth at low P by selecting for more whorls. And this is work under drought at our site in South Africa by Katie Barlow. Katie found in a population where we had variation between 1, 2, and 3 whorls in the same genetic background that no effect on growth when irrigated, but under drought stress, a 70% increase in growth associated with this trait, which, by the way, is under simple genetic control. So there's an example of finessing a trade-off between deep and shallow resources. Another option is to use genetic mixtures or multi-lines. So we have some shallow-rooted lines that are good for low phosphorus, deep-rooted lines that are good for drought, and we're actually planting a mixture of these lines, which thereby reduce competition between neighboring plants and also have a better range of soil resource capture as a stand or a field level. So this is what it would look like. These are recombinant inbred lines from the same parents, so they look very similar. The seeds look the same. The shoots look the same. So the farmer really doesn't have to be concerned about the fact that it's actually a genetic mixture, but it's actually 2 different root architectures in this multi-line. This is work of Amelia Henry, who's now at Erie in the Philippines. And so Amelia did this work on farm with small farmers in stressful conditions in Honduras. And you can see the multi-line yields are in red, and the single-line yields are in blue, the average of the 2-component single lines. And you can see in most environments, and overall, there was a significant yield gain from planting multi-lines as opposed to single lines. So I think this is a promising avenue for some of these root traits. Rather than trying to get one genotype that does everything, we combine genotypes in a field, so as a field, as a community, the system is more efficient. Another concern our stakeholders had about this technology was once we began showing we could increase phosphorus extraction from the soil, our sponsors and stakeholders were concerned that we would be degrading soil fertility over time. We're going to be mining the soil. We're taking phosphorus out of the soil, and in a situation where the farmers are not using fertilizer, we're going to, basically decrease soil fertility over time. Well, that was an interesting issue. It hadn't been researched. We thought that it was not going to be a problem because of this sort of thing here. This is a cartoon based on literature data. So in a current bean production system, assuming we have 1 metric ton of yield, which is twice the world average, in that 1 metric ton of yield, we have 2 kilograms of elemental phosphorus in the seed. So every year we're taking away 2 kilograms of phosphorus in the seed. But in these systems, erosion is the biggest loss of phosphorus. You might lose up to 100 kilograms of phosphorus per hectare per year in erosion. So we felt that by doubling growth and biomass and yield to 2 tons, now we're exporting 4 kilograms of phosphorus in the seed. So we're mining the soil, right? We're taking away 2 extra kilograms. But by having a bigger plant, we're protecting the soil from erosion and thereby reducing the biggest loss of phosphorus. And there'd be a net benefit in terms of phosphorus cycling in this system. In addition to benefits in terms of the farmer having more food and more income and these kinds of things, it might actually help conserve soil fertility. This was the theory. This, once again, was Amelia Henry's work, working with small farmers in Costa Rica. Amelia, she just rented fields from these farmers. You can see the steepness of the slopes they're farming on. And she installed these lysimeters to catch the runoff and measure the phosphorus coming off the field. Over a 2-year cycle of maize being maize, she was able to compare the phosphorus cycling in several sites such as this, comparing these conventional deep-rooted lines, which, as you can see, do not grow very well in this low phosphorus soil, compared to these new shallow-rooted lines, which grow better and protect the soil better. And Amelia found that she was able to reduce phosphorus runoff by 50 to 70% with these new shallow-rooted lines. So there seems to be actually a benefit for phosphorus retention in these systems. So what about Carioca? After some 25 years of effort, Carioca was the Brazilian landrace that nobody could beat back in 1990. Here it is as a check against some of these new materials. It's actually much worse than the new materials. This is Steve Beebe, leader of the CIAT Bean program. This is some of Steve's data. Here's Carioca. When you fertilize it, 2.5 tons. Without phosphorus fertilizer, 700 kilos, which is a very typical yield for Carioca. Some of these new lines selected to have good root traits. In this case, G1NA33 has triple the yield of Carioca, so over 2 tons without fertilizer. These two have double the yield without fertilizer. So through direct phenotypic selection, even though we've identified QTL for these traits, we haven't used molecular breeding. Through direct phenotypic selection, we've identified a number of these lines which the CIAT Bean program is using as parents in their crossing programs and therefore being deployed through Africa and Latin America. To push this technology forward more directly, various sponsors have supported our work in Africa to work directly with bean breeders. This is Celestina Joshua, who's the bean breeder of the government of Mozambique. This is Magalhães Miguel, a physiologist, Suarez Serinda, agronomist. They're all Penn State students and working there in Mozambique. So Celestina, using direct phenotypic selection for a couple of the traits I've shown you today and another one, has been able to develop lines that have 2 to 3 times the yield of the best performing materials that we had previously in multiple locations in Mozambique under drought and low soil fertility. So significant yield gains by direct phenotypic selection for root traits. Where Celestina, Mozambique is a very poor country. Celestina does not even have reliable access to electricity. So molecular breeding is really out of the question. So these sort of direct phenotypic selection procedures are very appropriate for her conditions. Suarez has shown that the new lines have much greater nitrogen fixation. They have substantially reduced soil erosion and better water infiltration. They perform well in maize intercrops, which is how beans are usually grown in Mozambique. And they utilize rock phosphate better, which is important because this is, if they have fertilizer access in Mozambique, it's going to be rock phosphate. So this is what I thought was a very successful program. We've increased yield. We have agroecological benefits. Our work is done, right? But our sponsors really aren't interested in those things. Our sponsors are interested in how we're affecting people with these new seeds. And so we're working with Jill Findice, a social scientist, who's now at the University of Missouri. The lines are being released starting last year. And she's conducting very thorough impact assessments and so on to see what's happening with this new technology. And it turns out that the impacts we're having are very dependent upon the seed type. This seed type is actually a Carioca seed type, the one I was telling you about before. That has no market value in Mozambique. So if it has no market value, it's under the control of the woman who usually grows the beans. And she will feed her children with this extra yield of Carioca, improved Carioca, which is very important because 70% of the households in Mozambique are food insecure. And this is the impact we were hoping to achieve. This type, the sugar type, is the common commercial class of bean seed in Mozambique. So because it has commercial value, it's taken by the man and he sells it in the market. What will the man do with the extra income? He will buy beer. This is just the person we happen to photograph. I'm not singling out this individual. Joe has done hundreds and hundreds of surveys, and this is the most common outcome of increased income for men, is the first thing they will do is buy beer. Now, it may seem to us that this is sort of a waste of income. The men will tell you that, no, this is important for the family because these people do not own their own land. And by drinking beer with the village chief, their family might have a better allocation of land in the upcoming season or something like this. So it's sort of a social capital. Okay? But now if we improve the yield of some of these very high-value grain types, such as this one, I have seen this grain type, the Kalima type, selling in large cities in Mozambique for $1.90 a pound, which is more than you pay for beans here in Iowa, probably. The average income in Mozambique is 70 cents a day. So this is a very high-value crop. If you have access to one of these urban markets and can grow these high-value seeds, you can make a lot of money. What will men do when they have a lot of money? They'll probably marry another wife. This is a polygamous culture, and so we interviewed individuals that had 6 or 7 wives. If they were successful farmers, they made more money and had more wives and more children. So it's kind of very different development outcomes. In one case, the women, in this case, are feeding their children with this class of beans. In this case, the men are expanding their families with this class of beans and drinking more beer. And so, of course, we don't want to be biased as foreigners in terms of telling these people what they should do or what's right and what's wrong. But we have an interest in not being biased towards just benefiting men or women for that matter. So what Jill has done is gone into these villages and mapped very carefully the spatial location of every household and understood who shares beans. It turns out, since these are very valuable products, they don't share bean seeds readily with their friends even. You'll only share bean seeds within a family and so on. And so she's looked at, these are large networks of relationships. This is just a very small segment of her data set. This is 4 families in a village, okay? And she's looking at the relationships in terms of where they get new bean seeds. There's no formal seed systems in this area. You cannot buy seed. You have to get it from somebody. So if we look at, in this particular case, the husband's networks are relatively more extensive because the husbands actually don't work on the farm. They don't take care of the children. So they're trading or drinking beer with their friends or something. So they actually have more extensive networks. The women are more likely to be localized around the compound because they're taking care of children and the plants. And so if a new bean is introduced that the men can sell and make more money and buy more beer, that information and that seed will travel readily through these networks. Whereas if a new bean is introduced that benefits the women's ability to feed their children, this technology is really not going to move very far in this system. So even if we make the various types of improved beans equally available to men and women, the women's preferences are not going to be moved through these communities as rapidly as the men's. So in response to this, Jill has had to develop kind of innovative seed dissemination technologies. How can we disseminate lots of seed to lots of people without circumventing the traditional tribal authority structures and without biasing ourselves towards men and women? So it's kind of an interesting challenge. In response to our successes in Africa, the McKnight Foundation asked us to work with soybean in South China, which is also a food security crop. South China is a low phosphorus area. And we did this work in collaboration with Professor Xiaolong Yan at South China Agricultural University, who tragically passed away from cancer several years ago. Basically, soybeans and beans are very similar plants in many ways. This is what soybeans look like, commercial soybeans in South China. And this was our idiotype. This was what we thought they should look like. They should have these stronger, shallower roots to get that phosphorus. Xiaolong's group, over a period of 8 years, using direct phenotypic selection for root traits, was able to select lines that had these shallow root traits. It was very successful. At this point, 15 of these phosphorus-efficient lines have been released in South China, in provinces inhabited by 300 million people. They're seeing an average yield gain of 20 to 30% in these low phosphorus soils. So this is not something that just works in common bean. I think the same strategy works in soybean. And of course, many of you here are interested in corn. We've also done this work in corn and seen a lot of the same biology in terms of phosphorus efficiency in corn. But phosphorus is important for corn production in Africa and Latin America. But I think a bigger issue with corn, of course, is water and nitrogen. It's a big plant, needs a lot of water and nitrogen. These strongly limit yield in developing nations. Drought's the biggest risk for corn production in the U.S. in the rich nations. Of course, nitrogen fertilizer is one of the principal production costs, and it's the primary energy cost, and it's the primary environmental cost of growing corn in the U.S. Amazingly, and the cost of nitrogen fertilizer has been increasing with energy costs. Amazingly, over half of our applied nitrogen in high input systems, such as here in Iowa, is not even taken up by the crop. It goes off into the environment, causing environmental pollution. So this seemed to be low-hanging fruit. If we could improve nitrogen capture and water capture, we could have a big impact both in rich and poor nations. And so this is what happens in Pennsylvania in the spring. This is a computer model, but it's parameterized from actual data from our field site in Pennsylvania. This is 42 days of maize growth. The root is growing, and that red is a pulse of nitrate moving down into the soil. Every time it rains, it pushes the nitrate down a little bit further. So it's kind of a race between leaching of nitrate and ability of the corn roots to capture it. And this is one reason, a principal reason we lose a lot of nitrogen fertilizer is the early spring rains wash it out before the roots can get it. So our idiotype for water and nitrogen acquisition is called steep, cheap, and deep. It's a variety of traits. Most of these are hypothetical, although if we're getting more and more data about some of these, I'll show some of it today. The idea is to go as deep as you can, as fast as you can, to catch that leaching resource before it disappears. So I'm going to show you a couple of examples of what we're talking about here. For a steep trait, meaning an architectural trait for nitrogen capture, I'll focus on optimal number of crown roots. Okay, what do I mean by this? There's lots of genetic variation for all these root traits in maize. This is the NAM 5000, and we're looking at the number of crown roots that we can see genotypes with as few as 5 and genotypes with as many as 35. So here you can see a genotype with very few crown roots, and here's very many crown roots. And our computer analysis indicates these are seminal roots here. The crown roots are these steeper ones coming out here. So this has 6 crown roots, and this has 36 crown roots. Our computer analysis indicates that you don't need that many crown roots. In terms of water and nitrogen acquisition, it's fine to have fewer crown roots. These are so close together that they're competing with each other, and the plant is wasting resources in building all these roots it doesn't need. So our idiotype was, let's reduce the number of crown roots and have a smaller burden of producing all these roots. So this is empirical work by Pattapong Sangwalai, who's now a professor in Thailand. This is our field site in South Africa. So Pattapong showed that rooting depth here measured as D95, which is the depth at which 95 percentile of roots reach, is related to crown roots. If we have fewer crown roots, we have significantly deeper roots in the field. If we have fewer crown roots, we have better capture of N15 that we apply deep in the soil. So we're getting better exploitation of deep nitrogen. If we have fewer crown roots, we have better dry weight. And if we have fewer crown roots, we double the yield under stress. Okay? So this seems to be a trait that's useful for nitrogen capture. In terms of cheap traits, what I'm talking about here are metabolic traits that affect the metabolic cost of soil exploration. So the first one I'm going to tell you about is RCA, that's root cortical parenchyma. This is what it looks like. Can you see this okay, or can we turn the lights down a little bit there, Jordan? Some of these movies are going to look better if it's a little dimmer, I think. So this is an actual section of a maize root that we've ablated with the laser. And you can see the green areas is air spaces that this is root cortical parenchyma. There's genetic variation for this trait. By converting living tissue into air space, it makes the roots much cheaper. You can see the natural genetic variation in maize. If we increase the parenchyma from 0 to 20% of the root cross-sectional area, we can actually cut root respiration in half. Here's the sort of genetic variation we see. These are unstressed plants, actually rills of the same population. And we use computer modeling to see how important this could be, so we know where parenchyma is in the maize root system. The hotter colors here is more parenchyma formation. We know how it affects respiration and nutrient content and other physiological processes. This is a physiological model called SimRoot. So we can use this model then to estimate what would the formation of parenchyma do for nutrient capture. So here we see a high and a low parenchyma maize line growing in a simulated low phosphorus soil. Otherwise, they're identical root systems. The only thing different is the parenchyma. And you can see over time the higher parenchyma lines ends up being bigger. And this is kind of an autocatalytic effect where every small segment of root is a little bit cheaper here, and so it grows a little bit further into the soil, catches a little more phosphorus, which results in more photosynthesis, and there's a feedback on root growth. So a small effect of changing the cost of root growth has a large effect on eventual root size. In fact, this is work of Jauke Postma, who's now in Jülich, Germany. Jauke's modeling suggested that as we go to more and more stressful conditions, as we go from 100% of normal yield down to like 10% where African farmers are, the benefit from parenchyma under nitrogen stress would actually improve growth by 60%. Similarly for phosphorus and potassium. Now, I admit that when I first saw these results, I didn't believe them. I thought that seemed like a very large benefit, 60% increase in growth just because of saving some root cells, the cost of root cells. Jauke's modeling also indicated that even at high input conditions like we have here in Iowa, parenchyma should reduce the need for nitrogen, phosphorus, and potassium. Now, 13% reduction in nitrogen application would be about $350 million a year for U.S. corn growers and significant environmental benefits. So that's a significant gain even at high input. Once again, coming back to Pattapong's work to verify these modeling results, this is in the field in South Africa and here in greenhouse mesocosms. He showed that lines with high and low parenchyma, comparing them under high and low nitrogen, the higher parenchyma lines had deeper roots in the greenhouse, deeper roots in the field. This is one of the field environments here in South Africa. He saw a 60% increase in growth under low nitrogen in the field in South Africa. In the greenhouse, a 60% increase in growth because of parenchyma under low nitrogen. In the field in Pennsylvania, which is not quite as stressful, a 33% increase in growth caused by parenchyma under low nitrogen. And a 60% increase in yield under low nitrogen in the field in Pennsylvania. So the idea of cheaper roots being better for nitrogen capture seemed to be working in the case of parenchyma. Parenchyma was also useful for drought tolerance. This is work of Jinming Zhu, who now works for Monsanto. We're using rainhouse shelters to create drought environments there in Pennsylvania. And you can see, in this case, comparing high and low parenchyma lines. This is high parenchyma lines, low parenchyma lines. The red bars are drought stressed. The blue bars are well watered. And you can see a substantially deeper roots under drought caused by parenchyma. The high parenchyma lines had better plant water status under drought. And they had actually 8-fold better yield under drought. This is what the ears look like. So this water-stressed plants are on the top. You can see the rills with parenchyma were looking pretty good compared to those without parenchyma. So this was interesting because it told us that the idea of looking for cheaper roots seemed to be a good idea, which suggested some other traits that people hadn't thought about that might make roots cheaper. This is a very simple one. There's genetic variation for the number of cortical cell files in maize roots. So here we have a genotype with many cell files, and here's one with few cell files. And of course, this one is more costly to build and maintain. And here we have genetic differences in cell size in maize roots. And so I'll be talking about these two traits now. So this is work of Joseph Chimungu in my lab. So here we have a genotype with only 6 cortical cell files. Here's one with 13 cortical cell files. And we actually have up to 18 cortical cell files. There's 300% variation for this trait in maize lines. So here's IBM lines in Pennsylvania. Here's land races in Malawi. And this variation is associated with the cost of roots. If we have fewer cell files, we can reduce root respiration in half. If we have fewer cell files, we can have deeper roots under drought. Fewer cell files give us better plant water status under drought. Fewer cell files gives us better growth under drought. And here in Pennsylvania, 2 years, and in Malawi, 2 years, it shows that lines with reduced cell files have better yield under drought. In this case, we have about a third increase in yield under drought. This is a doubling of yield under drought. In this case, a doubling. In this case, once again, about a 33%. So significant yield gains under drought associated with natural variation for this trait. Here's the other phene I was talking about, cell size. The idea being that larger cells are mostly vacuole. So they're cheaper to build and maintain than cytoplasm. We have significant variation for this trait in maize, about 10-fold variation. And when we compare, once again, we look at the physiology of these lines. As we increase cell size, we can cut root respiration in half. And just as we saw before with the other traits, if we have cheaper roots, increasing cell size, we have deeper roots under drought. We have better plant water status under drought. We have better biomass under drought. And we have, in this case, 2 seasons of doubling yield under drought associated with this trait. So, I think there's been a lot of research over the years on water use efficiency, transpiration efficiency, where we're looking at how leaves spend water to get carbon. But these results I'm showing you would suggest there's another way to look at this, which would be like a carbon use efficiency, which is how much carbon roots use to get water. So parenchyma, cortical cell file number, cortical cell size, these 3 traits I'm showing you, which have significant effects on drought tolerance, act through this mechanism, not through this mechanism. So I think there's a number of traits we can explore that might help us in terms of getting cheaper roots. So these lines are being evaluated and deployed in Malawi by Moses Muliro, the maize breeder in Malawi. And he's having very good success with small farmers and multi-locational trials with lines that have these cheaper root traits. Well, how do we phenotype root traits, which comes to the sort of subject that we're supposed to be focusing on today? We have developed this technique called shovelomics of simply, sending a graduate student out in the field with a shovel. Last year we went through about 25,000 plots from a variety of species. It takes us about 2 minutes per genotype once we get rolling. So this is from our site in South Africa. You can see here we've excavated some maize root crowns. Here's Eric Nord doing a visual scoring, and then we photograph them. And with the photograph, we're collaborating with folks at Georgia Tech who developed software with us called DIRT, digital imaging of root traits. So to take this photograph, and it's sort of something that African farmers or African breeders can use because you just upload the photographs online and the processing is done by computers at Georgia Tech. And then we're doing morphometric analysis to measure a variety of traits from these photographs. So for example, this is one of the traits we're looking at, angle of crown roots. You can see here again the NAM 5000, lots of variation for angles. Here's a shallow angled line, and here's a steep angled line. We've looked at 8 replicates of the Wisconsin Diversity Panel developed by Sean Kepler at University of Wisconsin. Over 4 years, that's about 500 entries. And this is work of Hannah Schneider, so we're not through with this analysis, but just to show you that even though we're digging up plants in the field, it's very quick and dirty, we are able to get genetic signals from this. In this particular case, here's a candidate gene that, according to Sean's gene atlas, is expressed in roots, and it's significant, and we know something about the biology of this in Arabidopsis. It's part of the auxin response pathway. So it seems reasonable as something that might actually be controlling root angle. For anatomical traits, we developed this technique called laser ablation tomography. Here's the laser here. Here's a little corn root sitting on the stage. It's about to be destroyed by the laser, and we're going to photograph it as we destroy it. This is what we see. This is a real-time image of a maize root being fed into that sheet of laser light, and it's being vaporized. And as it's being vaporized, we're photographing it. So what we end up with is a three-dimensional image of what that root looks like, including its anatomical features. And we can do 3D analysis, in this case of parenchyma we'll be seeing in yellow here, or xylem traits, which are also interesting, are in these blue and green colors. That image I just showed you took 60 seconds to acquire. So it's a very high throughput, powerful phenotyping platform for anatomical traits. Actually, we've recently begun looking at roots growing in soil using the laser. So this is a maize root growing in soil. We're ablating away the entire sample is being destroyed. We're just using the image analysis to keep the root here so you can see it. It's blue because it's a UV laser, so we're looking at ultraviolet fluorescence. And we're able to visualize mycorrhizas and other interesting things that we see in soil. So I think there's going to be a nice platform to look at what roots are doing at this scale of a couple of microns to a millimeter or so. Using that platform, using those images, we run them through RootScan, which are 2D image analysis software we developed to automatically capture root anatomical traits. And once again, without going into details about the GWAS, this is work of Patampong Sengwalai, just to show you what's possible to identify genes using this platform. Here's a candidate gene that, according to Sean's gene atlas, is expressed in the cortex where we would expect for parenchyma. This is root cortical parenchyma. In this particular case, this candidate gene is a transcription factor that is involved in programmed cell death, which is what you'd expect for something that was involved in parenchyma formation. So we're trying to chase this down, but I'm just showing this as an example that we actually can get genetic signals from these field-grown roots harvested in this kind of quick and dirty way. Now I've talked about several anatomical traits and architectural traits and how they might interact. There are so many that we're looking at. We're looking at about 30 different traits, and so the number of interactions is quite large. So here's a model then of tissue-level traits. This is a parenchyma we can see in here. So we're capable of modeling both anatomical. This is a structural functional model at the anatomical scale, and we have structural functional models at the architectural scale. So we can evaluate in silico how these traits might interact to affect fitness. And we see significant interactions. For example, here's 2 traits I've talked to you about today. Root cortical parenchyma is this trait. The number of nodal roots or crown roots is this trait. And here we have total biomass of the plants in silico. This is with different nitrogen levels. This is with different phosphorus levels. These 2 traits interact. If you're going to build more roots, it's more useful to have a parenchyma because it's important that those roots are less expensive. So we see an important fitness interaction between these 2 traits. If we increase the nitrogen level, the interaction changes. If we increase the phosphorus level, the interaction changes. And so we have a very complex phenome space of multiple traits interacting in important ways and their interactions themselves depend upon the environmental context. So in fact, we estimate that the scenarios of interest to us equal about 10 to the 23rd scenarios of different root phenotypes in different environments. There's 10 to the 23rd combinations. So using a powerful multi-objective genetic algorithm, this is work of Harini Rangarajan, she was able to plow through 10 to the 23rd possibilities using about 1 million hours of CPU time in December. And out of those 10 to the 23rd possibilities, pulled out 250,000 optima. So here we have the fitness landscape from low to high phosphorus, low to high nitrogen. And here's biomass of these plants. Each one of these scenarios is a complete root phenotype with architecture and anatomy in a particular soil environment. And you can see it's highly complex, and there's a lot of things we have to look at and analyze here. I'm showing you this to give you a feeling for how complex phenotyping or phenome analysis really is. I think for some of you who perhaps aren't physiologists, you might think, well, we're going to measure a bunch of plants and get this value and track down some gene. I think understanding the phenome is actually much more complex than that. So here's what we're trying to affect, and here's where a lot of us are beginning. And there are some people I've spoken with, influential people, who feel that this is all we need. We need a lot of genomic data, and we need a lot of yield data from multiple locations, and we'll be able to make progress through genomic selection. And I don't think that's right. I think that's very simplistic and naive. I think we're going to have to understand the phenome to really make progress and build better plants and make dramatic yield gains. We have to understand what these traits are, what their costs and benefits are, how they interact with each other in the environment, how we build integrated phenotypes in order to understand how these genetically controlled traits are actually affecting system performance. And I've been using, to alert people to the importance of thinking about this in a more rigorous way, I've gone back to a word that was first developed in 1925, phene. It's a word we don't use very much. Phene is to phenotype as gene is to genotype. And we all know how many words we have to describe the genome. We have a very rich vocabulary of all kinds of genetic control elements. There's hundreds of words to talk about things in there that's more than just genes. But when we talk about the phenome, we usually use the word trait, which isn't even a scientific term. Phene is a scientific term. And I think we're conflating in our minds a bunch of different things in the phenome that are quite distinct. I would argue that there are things called phenes, which are elementary elements. They're rudimentary elements of the phenome that affect function and interact with each other to affect function. Function then would be things like nutrient acquisition or carbon economy or water acquisition, which then goes into performance that we see in terms of rooting depth or biomass or yield. But there's a lot of steps between here and here. And I see that as an issue in developing phenotyping platforms. We can measure canopy temperature or something like this, but that's not a driving variable. That's a response variable. That's out here someplace. Even rooting depth, which you think, oh, that's helping me get a cooler canopy under drought. Rooting depth is not a phene in my definition. I've shown you today several very distinct physiological phenes with different genetic control that all affect rooting depth. So I think if we can be more precise about the way we think about phenes, we'll have an easier time identifying the genes that control them, and we'll have an easier time conceptualizing how we want to build a better plant. So the phenome bottleneck. The phenome is more important for plants and ecosystems than the genome, by definition. The phenome is what matters. The phenome is more complex than the genome. Some people think even nucleic acid sequence is a phenotype. RNA is a phenotype. So basically everything a plant does is in the phenome. The genome is just kind of like a little information component that affects the phenome. After some 30 years of massive public and private investment in genome research, our understanding of the genome far outstrips our understanding of the phenome. Emergent properties at each biological level of organization need to be understood if we're going to really have impact in real-world systems. I think it's naive to believe that understanding a gene is going to help us understand what's going to happen in a field in Mozambique without thinking about the biology that's in between. Refocusing of the support, expertise, infrastructure, and training capabilities of the U.S. plant biology community, especially here in the U.S., has driven some phenomic disciplines to the point of extinction. Europe, Australia, and China have taken leadership, which is good for them, but I think it's bad for us. They're really it's like the number of people who think about these things in a rigorous way is dwindling, and they're getting gray hair, and they're really not being replaced. In my opinion, the principal limitation to translating genomic information to agri-ecosystem level processes is a rigorous understanding of, and in the U.S., our capability to even research the plant phenome from cellular to multi-organismic scales. And this is especially true of the stuff like I've been talking with you about today, roots, rhizosphere, soils, which is highly complex, poorly understood. It requires expertise in plants, microbes, and soil science that very few teams encompass. So in summary, root architecture is important for soil resource acquisition. Topsoil foraging enhances phosphorus acquisition, while subsoil foraging enhances nitrogen and water acquisition in leaching environments. Several anatomical and architectural phenes affect the metabolic cost of soil exploration, which is an important component of soil foraging efficiency. Bean and soybean genotypes incorporating root phenes for superior phosphorus acquisition are being deployed today in Africa, Asia, and Latin America. The steep, cheap, and deep idiotype for water and nitrogen acquisition in maize appears promising. High-throughput phenotyping platforms are available for these traits, and numerous genetic leads have been identified. So what are the prospects for the second green revolution? Breeding for yield at low fertility has been successful. In the few cases it has been attempted. Aluminum tolerance is working in places like Brazil today. Phosphorus, as I'm talking about today, is more complex, but it is working. We're having breeding impact with phosphorus-efficient lines. Nitrogen is quite complex, but as I'm showing you today, we have some promising traits. It seems like we can make progress with nitrogen capture. Nobody, to my knowledge, on Earth is interested in breeding plants with better potassium, calcium, magnesium efficiency. These would be important traits for acid soils in developing countries. They should be physiologically very feasible to work with. Recent advances in root biology are making it feasible for the first time to design root systems for specific production environments. So we might have a different root idiotype for corn grown in Iowa than corn grown in Georgia, for example. And we're at a point where we have the tools we could actually develop those idiotypes and develop the phenotypes and test those ideas. In my opinion, the bottleneck at this stage is on the phenome and not on the genome side. We need more information about the long-term system impacts of this technology, but it should be benign. Note that we're not going to have resistance breakdown if we have a drought-tolerant or nitrogen-efficient line. Unlike disease or insect resistance, that's a permanent technology. It's not scale positive, meaning unlike the first green revolution, it doesn't just benefit rich people. A nitrogen-efficient corn line would be beneficial in Iowa just as much as it would be beneficial in Africa for different reasons. And I think the need to reduce pollution in the rich nations is going to drive some of this. I know that the KWS, a German-based European seed company, is quite interested in these issues to reduce the pollution that they're having in Europe from nitrogen. Certainly, as soils degrade, fertilizers become costlier, and the world population expands. The need for this kind of technology is only going to grow in years ahead. So this is the team that actually did all this work, and our sponsors. So thank you very much.
So we have some time now for questions, and make a quick reminder, if you could please either speak into that microphone or raise your hand and I'll bring this one to you so that our colleagues out in cyberspace can hear us. And also for those people in cyberspace, you have the possibility to ask questions. You need to go to our website, and there is a tab that has a little form for you to fill out questions, and we have somebody here monitoring that. So if anybody has any questions, please go ahead.
I have a couple questions, actually. One concerns cheap. As a physiologist, I wonder if there might be any association between cheap cells in roots and cheap cells in shoots that might lead to, for example, cavitation in shoots under high water use conditions or evapotranspiration conditions.
Yes, that's a good question. And, you know, an engineer, my son is an engineer, and engineers talk about the iron triangle. Fast, cheap, and good. You get to pick two. You can't have all three. So when we're building cheaper root systems, are they as good? Are they as sensitive, are they as resistant to insect attack, fungal attack, drying soils, or are there shoot trade-offs? So these are the questions. And we don't know. There's no literature about most of these traits I talked to you about today. So we're trying to figure that out. One of the reasons we do computer modeling and greenhouse work and field work in the U.S. and field work in Africa is so we might see if there's some big issue, it might jump out at us. And so far, you know, we're getting yield in the field under drought. We're not seeing big side effects. So, but we don't know. This has to be researched.
Okay. Thank you. My other question concerns automation of the shovelomics approach.
Yes.
Yeah.
Yes, that's a good question. I have a guest research appointment in Jülich. Some of you know Forschungszentrum Jülich, which is the German phenotyping center run by Uli Schur. And with those folks, they have a lot of engineering staff and so on. We're trying to actually automate shovelomics right now. So the idea is you drive through the field in a tractor, and you're lifting the roots, slicing them, blasting them with air and cleaning them, imaging them, using dirt then to extract images, using SimRoot to extrapolate from what you see to what you don't see. So the goal would be driving through the field at 3 miles an hour, collecting your root phenotypes. I think that's feasible.
Hi, Jonathan. Good presentation, as usual. I was wondering if you could speak a little bit to the heritability and the G by E of some of these root traits that you talk about. And I think one really relevant E to think about in the developing world is no-till versus conventional agriculture. And then on top of that, when we talk about the throughput and the automation, maybe you could speak a little bit to the cost per sample and the cost per unit genetic gain for some of these other things like laser ablation, for example. It looks really expensive. I just don't know.
No. Okay. Yes, so there's some good questions. The initial concern when we started this work many years ago working with bean, breeders would say, oh yeah, you'll see differences in root architecture, but then it'll change with every soil. It'll change every side of the hill. We have a different root system. So how robust are these differences? So that was one of the first things we looked at. And what we've seen, you know, like last year we worked on 5 continents. So we work in a lot of different soil environments. We do work in the greenhouse and so on. And these root traits are surprisingly robust. Any kind of normal agricultural soil that doesn't have huge rocks in it or something, these roots have a genetic program that they want to look a certain way, including plasticity. In about half the maize genotypes, about half the bean genotypes, we see that certain traits are plastic, meaning they respond to environmental conditions. That itself is under genetic control. It itself is a trait. And our idiotype is we don't want plastic roots. We want stubborn roots that do what we tell them and ignore other information. But that's a researchable question. So there is plasticity, but it is under genetic control. And in general, when we look at soils on different continents, all the places we work, we see the same phenotypes. So I think that might address your first question, right? Heritabilities for these traits from these shovelomics, sort of quick and dirty techniques, depending on the trait, range from 0.4 to 0.85. And you can see we're getting, you know, genetic signals out of a GWAS analysis, QTL analysis, and that sort of thing. Your question about cost. Of course, one of the reasons we do a lot of work in Africa is because we have cheap labor. So a different set of costs if we're working in the U.S. I estimate our shovelomics platforms cost us about $5 a plot. And of course, the idea would be, you know, when we're working with African breeders, that's their selection is their phenotyping the roots. In Iowa, you maybe would find markers and be doing, you know, molecular breeding or something with those, so you wouldn't have to continually dig up roots. I think the tractor-driven shovelomics is going to be obviously much, much cheaper. You asked about laser ablation. That's actually a very inexpensive method. The competing technology that people use is micro X-ray CT scanning. I don't know if you're familiar with that technology. Those cost like $1 million or more, run by PhDs. It's a very high-tech type of instrument that doesn't have the resolution that we have. This system, everything, the computer, the software, cost us $30,000 and is run by an undergraduate who's paid $9 an hour. So it is, everything we do is quick and dirty. We don't get into the really high-end stuff. So this is actually a very easy, even the soil sample I showed you, there was no treatment or anything. We just take a handful of soil, plop it on the stage, zap it with the laser, and a few minutes later you see what you see. So it's really not, there's not a real cost barrier to that, if that's what you were getting at. Did I answer all your questions? Okay. Yes, Jiaming.
Very nice talk, Jonathan.
Thank you.
I like your summary or the perspective on the importance of phenomics after the genomics problem has been solved. But when we look at the genomics, it was mainly driven by the human medicine genetics. There's a huge push to funding NIH and SF to do those genotyping. Once you get DNA extracted, it's still the same thing. Maybe there's other scientists can adapt to different species, but it's pretty much the same sequencing. But when you look at the phenomics, it's all different.
Yes.
And then where do you see that? I know that we're working with engineers within the image analysis, CT scans. Where do you see other emerging areas that we might leverage instead of, like we work with some survived physiologists because of the funding issue.
Right.
What are the other potential areas we could look into because it's going to be lots of different things.
Yes.
Instead of just DNAs. Yeah.
Well, that's a very good question. Thank you for that question. I think, I have found that there's tremendous untapped potential for collaboration with computer scientists, information technologists, and engineers. Just as we've seen a tremendous revolution in our ability to understand the genome, you all see in front, you have laptops in front of you, you know that we've seen a tremendous increase in our computing ability. So this fitness landscape I showed you, where we looked at 10 to the 23rd scenarios, 1 million hours of CPU time. That just was not possible 5 years ago. That was military level supercomputing, and now it costs me nothing. NSF just gives that time away. But there's not a lot of plant biologists, at least in the public sector, who have the computing expertise to tackle these problems and apply high-powered quantitative computing techniques to these issues of the phenome, which, as you understand, are extremely complex. So I think that's something that, for example, I see as a training gap there, too. When I get a grant, I'm not able to hire a postdoc who knows how to do anything about modeling and who knows about plants and soils. Those people don't exist. So I work with grad students and I train them, so they can do that because they don't exist. So that's, I think, an overall need that I see. But I think there's a more fundamental problem. And I think, Kendall and I were talking about this last night over dinner. There's a fixation in this country on genes. And I don't mean that, as any kind of criticism of geneticists, and it's just amazing. It's a remarkable achievement of the human mind, what we have done in understanding the genome. But as I was trying to argue here, the phenome itself is more important and more complicated, and we're kind of ignoring it. And I think the kind of things that I'm showing you are really obvious in retrospect. Air spaces are cheaper than living cells. Isn't that obvious? Thicker root is more expensive than thinner root. This is not rocket science. The reason nobody has written about this is because I don't think anybody's looking at these things. They're all looking at genes. So I think there's an untapped opportunity for the young, the grad students here and the next generation to really take the phenome seriously and treat it as a subject in its own right and not just a way to find genes. I think that's really the unexplored opportunity, is let's take a serious look at the phenome. How come you haven't heard about the word phen except for me? That word was invented in 1925. I think that's the low-hanging fruit, is let's just take a serious look at the phenome and think about, idiotypes again, and think about what we want crops to be rather than all the genetic control elements. I think the phenome should drive our work, and the genetics is a tool to get there and not vice versa, which is what I see nowadays, at least in the public sector. Everybody's excited about genes. The funding is for genes. The training's on genes. Who's thinking about the phenome, really? So I don't know if that answers your question, but yeah.
Have you found a value in formulated soil mixtures to reduce variability compared to using native soils? In some situations, anyway.
Yes, we can get much cleaner data, much higher heritabilities if we use controlled environment, sand culture, absolutely. That's, however, much costlier, especially when you're working with people like Sean Kepler who are throwing, 500, 1,000 lines at you saying, can you phenotype these? So we actually end up working a lot in the field because it's just so much more cost effective. We can evaluate 10,000, 15,000 plots in the field. We don't have the resources to look at 500 plants in the greenhouse. It's just a whole different level of, maybe Pioneer could do that, but I have as Gerald introduced me, my undergraduate degree is in soil science. There's a lot of different soils out there, and I think we ought to be looking at plants growing in the soils that we're interested in at least part of the time, and before we get too involved in some kind of artificial. So if we can do things in the field and it's in the real soil, why not, And that gets to the question about the G/E that we had too. Look at things in Iowa, look at things in Wisconsin, look at a few of your major soil types and see if you see differences.
So, sorry, unfortunately we're out of time for questions, guys. We have to get moving on to our next speaker so that everybody gets a fair amount of time. But Dr. Lynch will be around for the rest of the day, and we have some break time. So.